Duplex sequencing explained, using molecular barcoding to detect ultra-rare variants. Let's imagine you are examining a patient's genetic sequencing data. As you filter for variants, you notice one in a clinically meaningful gene. However, it appears at an exceptionally low variant allele frequency. It could be a real variant, or it could be nothing more than a sequencing error. Although next-generation sequencing is very powerful, it's not perfectly accurate. Most methods have an error rate of between 0.1 and 1%. This graph shows the frequency of variant calls for each base made by the sequencer, including calls stemming from sequencing errors at a rate of around 1%. The signals for high-frequency variants, such as germline heterozygous or homozygous variants, stand out clearly against the background error rate. But when the true biological variants are extremely rare, let's say appearing at a frequency of less than 1%, their signal falls into the same range as the error rate, making them indistinguishable from the background noise. This is the problem that duplex sequencing solves. It removes errors generated during the sequencing process and enables the confident detection of true, ultra-rare variants. In this webinar, you will learn about the duplex sequencing workflow, its advantages and limitations, and the key applications of this technology. The duplex sequencing workflow starts similarly to standard next-generation sequencing methods. First, purified DNA is fragmented, size-selected, and end-repaired. The fragments then undergo 3' adenine tailing and are ligated to adapters that contain unique molecular barcodes. The library is amplified, sequenced, and then processed for error correction. Let's walk through each step of this workflow. First, purified sample DNA is fragmented to prepare for sequencing. This can be done through sonication, which uses ultrasonic waves to break DNA. Next, beads are added to the test tube containing the sample DNA and placed against a magnet which attracts beads that are bound to large, poorly sheared DNA fragments. The supernatant will be kept while the beads are discarded. After size selection, the DNA undergoes end repair. DNA polymerases fill in 5' to 3' overhangs, while exonucleases trim 3' to 5' ends, and together they create blunt-ended fragments. Once blunted, a kinase adds a phosphate group to the 5' ends, preparing them for ligation. Next, the DNA is subject to 3' deoxyadenosine tailing. This enzymatic method adds a non-templated adenine to the 3' end of the blunt, double-stranded DNA target molecule. This is done to prepare fragments for ligation with adapters. The adapters are synthesized by annealing and extending 2 oligonucleotides at complementary regions. Each adapter molecule contains a 12-nucleotide randomized sequence called the duplex tag. The duplex tag is a unique molecular barcode that will label each DNA fragment. Each adapter also has a fixed nucleotide sequence, which is necessary for ligation. The fixed sequence contains a restriction site, which is cleaved by a restriction enzyme, creating a 3' deoxythymidine overhang that ligates to the 3' adenine overhang on the DNA fragment. Here, you can see the true variants highlighted in brown and black. Now, each DNA fragment is ligated to 2 adapters. This means that each fragment now has 2 unique barcode tags. We will call these the alpha tag and the beta tag. The fragments are then amplified using PCR, with each molecule being bound to flow cell adapter sequences, just as in standard next-generation sequencing methods. PCR amplification of the fragments occurs by extension from the 3' ends of the flow cell adapter sequences on each molecule. As PCR cycles continue, each strand-specific product is amplified, forming families that can be traced back to their original starting molecule by their unique tags. Although PCR errors can occur and propagate throughout the families, they will be removed later on during the error correction process. After amplification, the products are sequenced using standard next-generation sequencing. This will result in 2 types of read families being called. The forward strand will be captured by reads that are tagged in the alpha, beta orientation. The reverse strand will be captured by reads that are tagged in the beta, alpha orientation. Let's see how well you understand molecular barcoding so far. In duplex sequencing, 2 distinct molecular barcodes are appended to the ends of each DNA fragment. Why is this necessary? A, to increase amplification efficiency during PCR. B, to allow the DNA fragment to bind to the sequencing flow cell. C, to uniquely identify which strand of the original DNA duplex each read originates from, or D, to prevent sequence adapters from self-ligating. Take a moment to pause this video. The answer will be revealed shortly. The correct answer was C. Let's explore each option again. A was incorrect since PCR amplification will take place regardless of the tag sequences. B is incorrect because the flow cell adapters are what allow the DNA fragments to bind to the flow cell, not the barcoded tags. D is incorrect because the presence of distinct tags does not prevent ligation. C was the correct answer. By giving each strand a unique combination of two tags, alpha beta on one strand and beta alpha on its complementary strand, we can track the strand orientation and identify reads coming from each strand of the original DNA fragment. Continuing along with the duplex sequencing workflow, once all fragments are sequenced, bioinformatic processing of the data begins. The reads are aligned to the reference genome, and non-mapping reads are discarded. Reads sharing the same tags are grouped together to form duplicate tag families. At each position, the most common nucleotide among the reads is selected to form the single-strand consensus sequence. This will remove most random errors. However, the single-strand consensus sequence can still contain an error. If, for example, an error occurs very early during the PCR process, such as in the first amplification cycle, or if the DNA strand carried a damaged base, that error will appear in every read from the strand and will be included in the single-strand consensus sequence. After generating a consensus for each strand independently, the two single-strand consensus sequences are then paired and compared to form a duplex consensus sequence. This acts as a second validation step for true variants. Only variants present in both single-strand consensus sequences are kept, while variants that appear on just one single-strand consensus sequence, such as PCR errors, sequencing errors, or DNA damage, are excluded. Now, let's see if you can work through an example on your own. In this alpha-beta tag family, a green variant appears in 5 out of 5 reads, and a red variant appears in 4 out of 5 reads. In the beta-alpha tag family, the green variant appears in 5 out of 5 reads, and the blue and red variants appear in 2 out of 5 reads. Based on this, which sets of single-strand and duplex consensus sequences should be created? Pause the video to think, as the answer will be revealed shortly. The correct answer was option B. Let's break down why by looking at each option again. Option A is partly true. The single-strand consensus sequences are correct since the green and red variants occur in the majority of alpha-beta tag family reads, and the green variant occurs in the majority of beta-alpha tag family reads. However, the duplex sequence is incorrect because it wrongly incorporated the red variant, which was not present in both single-strand consensus sequences. In option C, the duplex consensus sequence was correct, but the single-strand consensus sequence for the beta-alpha tag family is wrong. The majority of reads in that family only contain the green variant, and so the single-strand consensus sequence should not include either the blue or red variants. That leaves us with option B, which has both the correct single-strand and duplex consensus sequences. That wraps up the duplex sequencing workflow and error correction process. In the next section, we'll explore the key benefits and limitations of duplex sequencing so you can see where it truly shines and where it might not be the best fit. The major benefit of duplex sequencing is its exceptional accuracy. Compared to standard sequencing methods, duplex sequencing reduces sequencing-derived errors from 1 in 1,000 to 1 in 10 million. Because errors are removed rather than mixed in with the data, the remaining signal more accurately reflects the original DNA molecules. By significantly lowering the background noise, duplex sequencing makes it possible to detect ultra-rare variants that would otherwise be nearly impossible to catch. Secondly, duplex sequencing filters out common artifacts and damage-related errors, such as those caused during sample preparation, as these typically only affect one strand. This would not be possible if a single-stranded consensus method was used instead. This feature also makes duplex sequencing especially valuable for formalin-fixed paraffin embedded samples, which are prone to oxidative damage due to the fixation process. Thirdly, duplex sequencing follows many of the same library preparation steps as standard next-generation sequencing protocols. The main difference occurs in the downstream data processing, which makes this method accessible to laboratories that are already performing next-generation sequencing. While duplex sequencing has significant benefits, it also has some limitations that may hinder a lab from fully adopting this technique. Firstly, generating a duplex consensus requires obtaining sufficient reads from both strands of each original molecule, which requires a very high raw sequencing depth of 10,000 or higher. This increases cost, computational load, and turnaround time. Secondly, duplex sequencing retains a smaller fraction of original DNA molecules compared to standard sequencing methods. A duplex consensus can only be formed when both strands of an original DNA molecule are successfully tagged, amplified, and sequenced. While this is fundamental to its error correction power, it also means that a large fraction of sequencing reads is discarded if one strand fails to amplify or doesn't have enough coverage. As a result, the overall yield is low and substantial data loss is common. Lastly, despite its superior accuracy, duplex sequencing is not completely artifact-free. A well-known issue involves end-repair artifacts generated during mechanical fragmentation in library preparation, which can result in false variant calls. Because of these challenges and the high resource demands, duplex sequencing has not yet been adopted in clinical diagnostics or in standard research pipelines. Now that we've covered the benefits and limitations of duplex sequencing, let's take a look at where this method is especially impactful. Duplex sequencing is most powerful in situations where biologically meaningful variants exist at extremely low frequencies. Let's walk through 4 key areas where this occurs. A clear use case would be detecting somatic or mosaic variants that occur in a small subset of cells. For example, identifying somatic variants in heterogeneous cancer tissues. Secondly, duplex sequencing has great value when studying mitochondrial genetics. Each cell contains thousands of mitochondrial genomes, and pathogenic variants often appear in just a small subset. Duplex sequencing can detect the rare, heteroplasmic variants that contribute to mitochondrial disorders or aging. The next key area is liquid biopsy and cell-free DNA analysis. Plasma contains cell-free DNA from all over the body, so DNA from a specific cell population of interest is greatly diluted by other DNA, and thus a highly sensitive detection method is required. Lastly, duplex sequencing can be used in mutagenesis and toxicity studies. For example, it can be used to detect DNA damage signatures after treatment with a new cancer drug, or to quantify the mutation burden in tissues after exposure to a toxin. Now let's look at a real-world example of duplex sequencing in residual disease monitoring in cancer. In a 2024 study, researchers followed 62 acute myeloid leukemia patients who were considered to be in remission after initial chemotherapy. They compared 2 methods for detecting residual disease: flow cytometry, which is the current gold standard for detecting residual cancer cells, and duplex sequencing, targeted towards a panel of leukemia-associated genes. Flow cytometry detected residual disease in 16% of patients, but duplex sequencing flagged residual disease in 35%. That's more than twice as many patients flagged. Duplex sequencing also proved to have much higher predictive power than flow cytometry. Among patients where duplex sequencing found signs of disease, 68% eventually relapsed. Among those with no detectable variants, only 13% did. This level of precision is extremely valuable. If DNA variants indicative of leukemia are detectable, doctors can intervene early and intensify therapy before a full relapse occurs. And if no abnormalities are detected, patients can avoid unnecessary overtreatment and gain a real peace of mind. This case showed the power of using the right method for the right question. Now it's your turn. Based on what you've learned, in which of the following situations could it be appropriate to use duplex sequencing? There can be multiple correct answers. A. Detecting pathogenic germline variants in patients with an ultra-rare disorder. B. Detecting low-frequency somatic mutations in heterogeneous cancer samples. C. Quantifying aging-associated mitochondrial DNA variants across tissues. D. Diagnosing overgrowth disorders caused by mosaic PIK3CA variants. Pause the video to think, as the answer will be revealed shortly. Options B, C, and D are all correct. They're all examples of situations where biologically meaningful variants are present at very low frequencies, requiring a highly sensitive detection method such as duplex sequencing. Option A was incorrect. The wording is a bit tricky. Although the prevalence of the disorder may be rare, the frequency of germline variants is not. A heterozygous germline variant would be present at an allele frequency of around 50%, which is much higher than necessary for this highly sensitive error correction method. Looking across all of these diverse applications, one theme is clear. Duplex sequencing enables us to confidently identify ultra-rare genetic changes that used to be just beyond our reach, expanding what is possible in the field of genomics. Thank you for watching our webinar.